On May 16, 2025, the FDA cleared the very first blood test to evaluate amyloid plaques in the human brain. This arrives against the backdrop of a massive and growing crisis, with Alzheimer's prevalence in the United States alone projected to hit 13.8 million people by 2060. Before this approval, getting a definitive look at underlying amyloid pathology meant either spending up to $8,000 on a PET scan or undergoing a painful lumbar puncture to collect spinal fluid. The newly cleared test measures a specific ratio of proteins in plasma, p-tau-217 to A-beta-42. The pivotal studies were impressive, posting predictive values well over 90%. But clinical reality is messy. When deployed, roughly 20% of individuals still land in an indeterminate diagnostic zone. The issue lies in the structure of the test itself. A single mathematical ratio lacks the biological context to distinguish between Alzheimer's disease and other conditions that cause tau pathology or mixed dementias. If we are going to screen populations effectively, relying on a simple binary threshold isn't enough. Diagnostics must capture multiple dimensions of the brain's environment at once. That is the exact problem a framework called Virtual Spectral Decomposition, or VSD, is designed to solve. Instead of measuring a single protein ratio, VSD treats a standard venipuncture blood sample like a spectrum of light, decomposing the fluid profile into a series of distinct biologically interpretable channels. The model utilizes 4 specific plasma biomarkers: p-tau-217, the A-beta-42-40 ratio, neurofilament light chain, or NFL, and GFAP. Each of those markers maps directly to an independent biological axis: tau phosphorylation, amyloid clearance, neurodegeneration, and astrocytic activation. But building an accurate diagnostic tool using these 4 channels led to a completely counterintuitive mathematical requirement. The algorithm actually relies on evidence of severe brain damage as proof against an Alzheimer's diagnosis. To achieve true precision, the diagnostic architecture has to be engineered with explicit disease-exclusion logic. The immediate challenge in combining these specific biomarkers is that they exist in entirely different concentration scales within the blood. The VSD pipeline solves this by running the raw data through a mathematical pre-processing engine. First, it uses z-score normalization to standardize raw plasma concentrations, mapping every individual patient's data relative to the broader population. Next, it applies a directional alignment factor. Because markers like the A-beta ratio actually decrease as the disease progresses, the algorithm flips them so that all indicators point in the same positive direction toward pathology. Those standardized values are then compressed using a sigmoid curve, bounding the data into clean channel activations, ranging strictly between 0 and 1. Finally, a logistic regression engine reads across those normalized activations and assigns a mathematical weight to each channel. To ensure accuracy, researchers rigorously calibrated these weights using ground truth amyloid PET scans from 1,139 participants in the Alzheimer's Disease Neuroimaging Initiative. Through this strict pipeline, chaotic biological measurements are systematically translated into an interpretable multi-channel spectrum. This chart shows the specific beta coefficients, or weights, generated by the trained logistic regression model for the 4 VSD channels. As expected, primary indicators drive positive diagnosis. p-tau-217 receives a massive plus 5.4 weight, and the amyloid ratio carries plus 3.2. But look at NFL. Despite being the gold standard biomarker for neuronal injury, it receives a negative minus 1.1 weight. The math reflects a clinical reality. If a patient shows highly elevated NFL, but lacks coupled markers for amyloid and tau pathology, they do not have Alzheimer's. An isolated spike in this neurodegeneration channel points firmly to alternative conditions like frontotemporal dementia, vascular dementia, or traumatic brain injury. So if a patient's blood profile enters the system with severe brain injury markers but low Alzheimer's proteins, the algorithm actively penalizes their Alzheimer's probability score, dragging it down into a rejection. This negative weighting acts as an explicit disease-exclusion mechanism, allowing the model to actively recognize and rule out confounding dementias. This raises a persistent biological puzzle. Why does p-tau-217 carry a dominant plus 5.4 weight, vastly outperforming the blood markers that directly measure amyloid metabolism. To answer that, we have to look closely at a massive cerebral spinal fluid proteomics dataset from the Cruchaga lab, which analyzed 7,008 distinct proteins to map the precise molecular footprint of the disease. Out of those thousands of proteins, the analysis revealed that only a tiny fraction, exactly 17, track amyloid pathology independently of tau. Compare that to the massive cluster on the other side. 826 proteins specifically track tau and neurodegeneration. That is a staggering 49-to-1 asymmetry. The biological implication here is clear. Neuronal injury and tau pathology generate a massive, highly reactive proteomic footprint that floods the surrounding fluid. In contrast, the actual deposition of amyloid plaques is biologically quiet. It barely registers in the fluid environment. So p-tau217 dominates the blood test model not because it's a direct measure of plaques, but because it acts as a massive biological megaphone, amplifying the otherwise quiet signal of amyloid pathology. Understanding these channels allows us to look past a single Alzheimer's test toward a broader architecture for multi-disease screening from one specimen. The VSD framework proposes a hierarchical routing layer called a dendritic gate, taking inspiration from how dendritic cells integrate signals in the immune system. In this setup, the data from one blood draw is split and processed simultaneously through multiple disease-specific analytical modules. We already saw how the NFL channel functions in the Alzheimer's module, utilizing a negative weight to act as an exclusion signal. But if you take that exact same NFL signal and route it into a Parkinson's disease module, the math inverts, assigning it a heavily positive weight. The underlying biology demands this flip. In synucleinopathies like Parkinson's, neurodegeneration isn't a confounding variable. It is the primary indicator of the disease. By routing channels through these parallel networks, the VSD architecture transforms a standard blood test from a static query into a dynamic cross-disease processor. This chart visualizes the clinical validation of the 4-channel VSD system, mapping its accuracy directly against ground truth amyloid PET imaging. At its optimal operating point, the algorithm achieves an 89.7% sensitivity and a 90.8% negative predictive value. Statistically, the area under the curve bumps from 0.888 for p-tau alone up to 0.900 for the full model. It's a mathematically modest increase, but chasing raw statistical improvements misses the point. The true clinical value of this framework lies entirely in the interpretability of its biological channels. Unlike a binary ratio, a physician can inspect the VSD channel activations to trace the mathematical logic of why the test produced its final result. A black box machine learning model could hit the exact same accuracy numbers, but it would provide zero testable mechanistic insights to the clinician. By replacing opaque ratios with a transparent, multi-channel architecture, VSD provides the mechanistic clarity needed to turn complex blood biology into actionable clinical decisions.